As Sindhu said, I've been here at the University of Washington for a long time. I'm a hematologist. I spend most of my time doing research. I still am on the clinical service. I happen to be on the leukemia service right now, but I just do that for just a very few weeks during the year. But I thought I would give you just a very brief perspective on how I've kind of come to working in various areas of research, gene therapy for a while, stem cells, still working on stem cells to some extent, but I've really become kind of enthralled with the notion of working in cancer. And I'll give you a little bit of a background about why that is and why I think the opportunities are so amazing and how I happen to land in this area. So really I guess, premise that it might make sense to become interested in doing something big came from initial work that was that came out of the work of this person, Jejun Duan, who is a who at the time was a research scientist in my lab, amazing molecular biologist, and Jejun, and in talking with Jejun, we became interested in the question of whether you could localize a gene within the nucleus of a cell, and whether it would be possible to see whether the state of a cell could be associated with the change of the position of any given gene within the nucleus. And when I asked Jejun to work on this, he converted this seemingly initial simple question into this notion of trying to understand where all of the genes are in the nucleus of a cell. The notion that you could think of the genome as being a plate, you know, we have chromosomes, 46 chromosomes, and we know where every gene is along every chromosome. That's what the Human Genome Project taught us, obviously. But chromosomes, you could think of them like strands of spaghetti out of a box. You could think of 46 strands of spaghetti. But chromosomes aren't really like spaghetti out of the box, they're more like spaghetti on the plate. But it's not completely random that there is an organization of genes within the genome, so that you could think of, so based on what Jejun did, you could think of a gene and trying to understand where it is within the nucleus of a cell, not only with respect to where its linear neighbors were along the chromosome, but you could think of it in 3 dimensions. So you could think of a genome as a 3-dimensional space, and you could begin to try to understand where a gene's nearest neighbors were in 3 dimensions. And the idea of this Google, analogous to Google Earth, this notion of Google genome. And could you imagine looking at a genome in 3 dimensions, swooping in and looking at the location of any given gene and understanding what its nearest neighbors were in 3 dimensions. Now, I have no business being involved in this type of project. That's really the point of this, that this was really an innovation of Jejun Duan, and he developed a way to characterize long-range chromosomal interactions on a genome-wide scale. And that was all Jejun. When it looked like that this was starting to work, we went to see Bill Noble, who's a professor in the Department of Genome Sciences and a computational guy, and Mirela Andronescu was a graduate student, or at that time a postdoc in Bill's lab, who knew how to make 3-dimensional structures from high-dimensional next-generation sequencing data that was initially applied to RNA structure. We then met with Jay Shinduri, who's one of the developers of next-generation sequencing, who became interested in the project. And it became clear that trying to characterize the 3-dimensional architecture of a mammalian genome was just too vast a problem. The genome's too large, and that we should focus our efforts on yeast. And so then we went to see Stan Fields, who's the developer of the yeast 2 hybrid system, and Kevin Schutz was a graduate student in his lab. And through a process that took about 2 years of meetings, weekly meetings, hour-and-a-half meetings with this group of people, we were able to construct a three-dimensional model of the yeast genome that shows, it's kind of hard to see here, what you see in the green is chromosome 12 looping out and going through the nucleolus and then coming back the other way. But this led to a paper where I think the real point of this is that the title of this paper that was published in Nature is a three-dimensional model of the yeast genome. I know very little about computers. I know where the on button is on my computer. I know nothing about yeast. And the idea that I would be involved in this project is completely counterintuitive. But for me, the lesson was that if you can find ways of working across very broad areas of expertise where people have completely different skill sets, that if one can navigate those between those areas of expertise and effectively coalesce them, then you can do amazing things that no individual could do or no individual lab could do. And so it was in that context then that I became, with the influence of my wife, so this is my wife, Sibel. She's an oncologist in Puyallup, kind of a very busy community oncologist. But it was in discussions with Sibel and going with her to some meetings, cancer meetings, that I became convinced that there are massive opportunities in cancer that aren't being adequately addressed. And the way that I think about it as a physician, scientist, is the notion of what it means to do our best. You know, what is your best? What does it mean to do your best? And I'm getting to be an old guy, so I came to Seattle in 1989. And the reason that I came here was because of this procedure that known as stem cell transplantation, bone marrow transplantation, where you, everyone here knows, you take a patient with leukemia, typically, push them to the very brink of death by giving them as much chemotherapy as they can possibly tolerate, and then rescue them with someone else's bone marrow. It's a very dramatic procedure. And I think that, and I think that really accurately fulfilled the 1989 definition of what it meant to do our best. But I would argue that today's definition of doing our best probably means something very different from that. And I would imagine, I would contend that perhaps no cancer patient is receiving what we could call our collective best today. And I should have changed this slide to reflect what was happening in this year, 2013. This is, but I think today's definition of doing our best requires that we meet a challenge that's shown in this graph, where if you look at the difference between exponential growth and linear growth, that technology is improving on an exponential scale. Yet the manner in which it's being applied to clinical care is still quite linear, quite similar to the same techniques that we've used for the last 20 or 30 years, with, I would argue, modest modifications. And I think the grand challenge of our time is to find a way to close that gap. And that's the question: how can we close that gap? And I think essentially the problem is distilled here, where I think there really is a growing tension between these dramatic and amazing advances in technology and the manner in which they're applied to the treatment of patients with cancer. And of course, the question is, can we do better? And this is just a graph that people will be more familiar with here, that shows the dramatic decline in DNA sequence costs relative to Moore's law that's used for the semiconductor industry for the last 3 decades. And obviously the decline in the cost to sequence a base of DNA has just fallen in a breathtaking manner. Obviously computational power continues to improve, and could there be opportunities that arise from that would require that we do things in fundamentally different ways from the way we do things now. What we know from DNA sequencing now, so far, is that every cancer's unique. And this graph comes from a review that that came out of Larry Loeb's group several years ago, showing on the top graph all the mutational profile of all colorectal cancers. And each peak here reflects an intersection between a chromosome and the percentage on this axis, the percentage of tumors that exhibit any given mutation. And the observation is, as everyone here knows, is that recurrent mutations in cancer, even the most abundant recurrent mutations, are rare. And the vast majority of cancers contain a preponderance of other mutations that are extremely rare in other individuals. And every cancer has mutations that are unique to that cancer. And whether they're driver or passenger mutations can be very difficult to sort out sometimes. So the top graph shows the heterogeneity in the mutational content of cancers across individuals, and the bottom graph shows the heterogeneity that occurs among cancers within an individual, so that we all in this room will recognize that even within an individual with cancer, there's a dramatic heterogeneity of the cancer depending on which site you biopsy. Cancers evolve. There are these evolving entities within individuals. And so the bottom line message of this slide is that every cancer's unique. And I think part of the challenge that we face today is that fact isn't adequately appreciated. I think the implications of this are profound and fundamental. And I think they really require that we think very carefully about how we try to tackle this problem. So, you know, the way, if you think about it, for example, in breast cancer, how many different kinds of breast cancer are there? And if you could think of it as if we go to QFC and we buy every fruit and vegetable that we could find, you know, how many are there? Maybe there are 30, maybe there are 40. And maybe if you want to do a randomized trial and you have enough patients in your randomized trial, let's say a couple thousand, maybe 4,000, all of those different subgroups will even out. Maybe you can even out the differences just by generating trials with very large numbers of patients. But what if the number of types of breast cancer that there are really is more like every item you could buy in Manhattan? And there is no number of patients that you could enroll into a clinical trial that would ever account for that type of heterogeneity. So we know for sure, it's inarguable that every cancer is unique. But our current clinical trial designs don't account for this fact. And we continue to treat cancer as if it were a black box, as if it's this immensely complicated entity, which it is, of course, but so complicated that it defies our ability to understand it really and to understand what's happening within individual patients with cancer. And so the way that we proceed then is to treat an individual cancer patient as if they were a black box, as if the disease that they contain is a black box. I'm on the leukemia service now, so I know we still do this. With, you know, greater than 20,000 genes, greater than 100,000 different RNA isoforms, I mean, immense complexity. And so the way we handle this is we take that one cancer patient, we find now many cancer patients, we treat them roughly Similarly, within each group, we compare two different treatments, the experimental treatment to the standard treatment, and we pick a winner. And if you happen, let's imagine that group A, so patients who received the experimental treatment, let's imagine 40% of them responded. And let's imagine that with the standard treatment, only 20% responded. And you could say, boy, you know, I hope I get randomized to the experimental treatment, but it's almost guaranteed that there were patients who enrolled in the experimental treatment, in the treatment that worked better for the population, that did not respond to that treatment, that would have responded if they had been assigned to the other group. And we have no way of capturing those episodes. And so I think this black box mentality becomes increasingly difficult to justify in the face of increasing knowledge. And this is highlighted by a specific case where there's been dramatic advances just in the last very few years that really, I think, highlight the difference between what technology can offer and the manner in which we test these things clinically. And that's shown here with the BRAF mutation, so a mutation that occurs in about 60% of patients with melanoma that turns the MAP kinase signaling pathway on fire. It upregulates the MAP kinase pathway by approximately 500-fold. And it's inhibited by a drug that now is FDA approved that has dramatic, albeit transient, efficacy in patients with melanoma. And so there was a lot of attention given to the investigation of this BRAF inhibitor in a clinical trial that's described here and was described in the New York Times several years ago, that described two cousins. And I don't know how many people have seen this, but this received at the time an awful lot of attention, where there were two cousins who both had metastatic melanoma, both young guys, and they both had the BRAF mutation, the V600E BRAF mutation. And I'll just read a few passages here. So growing up in California's rural Central Valley, the two cousins spent summers racing dirt bikes and Christmases at their grandmother on the coast. Endowed with a similar brash charm, they bought each other matching hard hats and sought ironworking jobs together. They showed a love for the rush that comes with hanging steel at dizzying heights and a knack for collecting speeding tickets. And then last year, they learned that a lethal skin cancer, melanoma, was spreading rapidly through their body, and they found themselves with a shared chance of benefiting from a recent medical breakthrough, because only months before a new drug had shown that it could safely slow the cancer's progression in certain patients. Both cousins had the type of tumor that was almost sure to respond, and major cancer centers were enrolling patients for that last crucial test that regulators required to consider it for approval for sale. And so one cousin says, "Dude, you have to get on these super pills," Thomas McLaughlin, then 24, whose melanoma was diagnosed first, told his cousin. Mr. McLaughlin's tumors had stopped growing soon after starting the pills. But when Mr. Ryan, 22, was admitted to the trial in May, he was assigned by computer lottery to what was known as a control arm. Instead of the pills, he was to get infusions of the chemotherapy drug that has been notoriously ineffective recourse for treating melanoma for 30 years, which I think was DTIC. And then describes what happened next. On the morning of May 12th, Mr. Ryan and his mother drove to UCLA. The cancer had spread through his body, yet that weekend his family was filled with hope. His doctor had found that the same gene mutation that his cousin had was in one of Mr. Ryan's tumors, and he was finally eligible for the trial. But the computer made its assignment the following Tuesday, making sure that he would not be getting his cousin's super pills. Mr. Ryan's mother picked up the call while her son was undergoing radiation for the tumor on his spine, and he was on oxygen. "I'm sorry," the doctor said as she cried into the phone. "There must be someone higher up she could talk to," she said, but there was not. It was completely random. No one could change it. And 2 weeks later, at his cousin's funeral in mid-June, Mr. McLaughlin placed Mr. Ryan's hard hat in his coffin and helped carry it to the grave. David Fisher said, "Many of my colleagues who are outstanding clinical investigators have been able to convince themselves that this is a fair thing to do. My personal view is it's nuts. I don't know of anyone who hasn't shuddered at the concept that we can't let patients on the control arm cross over because we need to let them die earlier to prove this point." And so I think this is, very clearly, I think, highlights a conflict between knowledge and the way that we've been doing things traditionally in cancer treatment. And I would argue that the future of what we need to do has to find fundamentally different approaches to trying to forward progress in cancer treatment. And I think a glimpse of that future could come from a study that many people here will be aware of, that was done by this guy, Mike Snyder, who's a professor at Stanford. And he published a paper this past March of 2012 in Cell, that I think described a very interesting approach that I think could be the part of the way toward the future in cancer. And so the way Mike describes this is he was recruited from Yale to come to Stanford. He got a lot of money, and he thought, I need to try to figure out how we're going to make personalized medicine a reality. How are we going to use all of this sequencing data to do any good for patients? And he's a PhD. He doesn't know much about human subjects, so he thought, I need to find a reliable patient, somebody that will come in for their blood draws when they're supposed to. He said, well, I'll do that. I'll just do this on me. And so he sequenced his genome using various platforms, various technologies, to an incredibly high level of resolution. So he had extraordinary confidence for each base of his genome that could be sequenced. And then he sequenced his mom's genome, so he could tell, okay, which of his, which alleles were paternally inherited and which were maternally inherited. And then he did RNA sequencing, and he looked at RNA editing events and allele-specific expression. And then over the next 400 days, he had his blood drawn 20 times. And he used his peripheral blood mononuclear cells to look at RNA sequencing and some antibody profiling, immune-related antibody profiling. And in the initial sequencing of his genome, one thing that was apparent that was completely unexpected was that he had an increased risk for cataracts, glaucoma. And that wasn't unexpected because there was a family history of glaucoma. But the other thing that he appeared to be at high risk of was the development of type 2 diabetes. And that was a surprise because type 2 diabetes really didn't run very heavily in his family, and he did not look like a guy destined to get type 2 diabetes. He was very fit, exercise fanatic, looked like the last guy that you'd expect to get type 2 diabetes. And essentially what happened was he did this integrative analysis of all of the omic profiles from his transcriptome, his proteome, his genome. And in monitoring his RNA-seq gene expression profile from his peripheral blood lymphocytes, he had 2 viral infections And the first viral, and then he really monitored himself a lot around those 2 viral infections. The first viral infection, you could see the gene expression profile from his peripheral blood mononuclear cells go haywire. And then as he gets better from the viral infection, it kind of returns back to normal, something close to what was for him his baseline. But then with a second viral infection, an RSV infection, some months later, his gene expression profile was dramatically altered, and then it didn't go back to normal. It was stuck in an abnormal state. Despite clearance of the viral infection, his gene expression profile had fundamentally changed. And so they started to look at all of the data that came from this, and they're trying to figure out, gosh, what changed? And they saw very significant changes in glucose metabolism and in insulin-related signaling pathways. And he goes to the doctor, and he finds that he's got diabetes. He's got type 2 diabetes. So he exercises like crazy, he loses 5 pounds, his RNA-seq profile doesn't change, he loses another 10 pounds, and now is really thin, and finally his gene expression profile goes back to normal, what for him was normal. And so when Mike gives this talk, he says, there's a guy in the back of the audience who raises his hand and he says, Mike, great talk, but I didn't learn anything from your talk. Yeah, it's an anecdote. And Mike's response is, it is an anecdote, but it's an anecdote with 3 billion data points. And I think how do we, and I think really the challenge is, how do we go from anecdotal evidence that we see in individuals to defining and perhaps arriving at new definitions of causality? I think that's really one of the major areas that will change a lot in medicine in coming years. So toward this end, I've had a couple of things that I've written or co-authored in the last year describing how we might try to apply similar approaches to addressing cancer treatment. And could similar approaches potentially be used in patients with cancer to try to understand what's going on in patients with cancer? So about 2 years ago, a little bit more than 2 years ago, I co-founded the not-for-profit organization that Sindhu mentioned, Partners in Personal Oncology. And I co-founded this organization with a neurosurgeon at Swedish Hospital, Greg Foltz. We have a very excellent and expanding group of people that have associated themselves with this organization. And the idea behind Partners in Personal Oncology is shown here in this cycle, but perhaps let me explain it first. So the idea behind Partners in Personal Oncology is twofold. First, the first idea is to fuse research and clinical care. Leave no gap between what a research lab can offer and what's made available to a cancer patient. So the idea is to have patients who are highly motivated to be the subjects of an unprecedented scientific investigation, where we would try to comprehensively characterize what's going on in their tumor, collect millions of data points. So if the tumor is present at multiple sites, biopsy the tumor at multiple sites, process it in a way that you have confidence in so that you don't perturb patterns of gene expression or the phosphoproteome, so you know that it's only seconds between the tumor being in the patient and the tumor being definitively stored. So biopsy the tumor at multiple sites, deeply characterize what's going on, do omic profiling on all of those bits, and then find the best people that you can find to look at this data, try to integrate it into some understanding of what's going on in this patient's cancer. And the obvious enormous challenge here is when you have millions of data points, there will be thousands, if not tens of thousands, of very plausible ways that you can put that data together, the vast majority of which are wrong. And so how do you find the true solutions out of the vast noise of potential false solutions? And so the idea here is that you take all of this information from a patient, you find the best people that you can find to work on it, they make a recommendation for treatment, and that recommendation is a recommendation that attempts to interfere with what we think is the key driver or the key drivers of that tumor. So the idea is that researchers can test their best ideas not in a lab animal or a petri dish, but they can test their best ideas in this human being who's so desperately in need of their best ideas. And so I think this aligns the interests of researchers and patients in a way that doesn't exist today, where researchers can test their best ideas in a patient, and the patient can be the beneficiary of what I believe would amount to today's definition of doing our best. Now, the key aspect of this is total transparency about how absolutely primitive our ability is to bring this data together in a way that accurately depicts what's going on in that patient's cancer. It's guaranteed that our predictions will be wrong more often than they'll be right initially. And I would not want to be held to the standard of doing better initially than an oncologist's guess. But the idea behind this is to do our best for a patient at any given moment in time, and then use that patient's response to our recommendation as a means for learning. So recommend a treatment, get a patient on that treatment, test in that patient whether our recommendation worked or not. If the tumor didn't respond, or if the tumor responds and then comes back, rebiopsy the tumor again comprehensively, repeat this cycle, come with our next best shot, make that recommendation in the patient, and collect this data over time in intensively monitored patients. And you can imagine this database that, with an enormous amount of information from patients before and after interventions that are designed to help them based on the best available knowledge and advice that we have at any given moment in time. And so, that's the first idea behind Partners in Personal Oncology. The second idea is the notion of a distributed network. So distributed with regard to the notion that we want to be able to get advice, expertise, technology, irrespective of where it may reside. We don't care where a person is. If they have anything that could be of benefit to a cancer patient, we want to gain access to that. But distributed also with respect to the notion that as cancer is increasingly recognized as an information problem. I mean, you could think of a cancer patient, you could convert a cancer patient into millions of data points. The challenge is how do you put them together in an accurate understanding of what's going on. But as cancer becomes an information problem, then much of the value that could be delivered to a cancer patient could be delivered through FedEx and the internet. And so that a cancer patient in rural Montana could potentially gain access to the same type of world-class expertise that a patient in Manhattan or Seattle could have. And so those are the two ideas. And this idea of all for one, meaning bringing the world's expertise to bear on the individual, and then one for all, using that individual's experience to try to add to our collective understanding of how cancer works. And so this is, working on, over the course of a couple of years, working to build a lot of infrastructure that would be necessary to make this succeed. And just a first hint of how this could work came from a patient of my wife's in Puyallup who had a gastrointestinal stromal tumor. And so this is just to very briefly recount his history. He was diagnosed in 2002 at the age of 44. He had a resection of a pelvic mass. There was tissue left behind. He was put on Gleevec. That worked for a few years. In 2006, it recurred, and he was switched to sunitinib. He responded in 2008. It recurred. He was switched to another receptor tyrosine kinase inhibitor, nilotinib. It worked. But then by July of 2009, his tumor was growing despite that treatment. My wife tried to get him switched to the latest TK inhibitor at the time, sorafenib, and the insurance company refused. But then, so she discharged him to home for home hospice. 5 days after he was at home to die, the insurance company shipped him the sorafenib. He took it, he responded again, was hanging Christmas lights by December of that year. But by the following year, the fall of the following year, his CT showed an interesting pattern where he had stable disease in the pelvis, stable in size, but what appeared to be growing disease in the liver. And so this seemed like a potentially interesting opportunity where we could try to understand what's going on with the growing disease by trying to understand the differences between growing tumor and stable tumor. And so that led to the notion of why don't we biopsy both sites, try to understand what the differences are, and could we, in so doing, find a vulnerability that would allow us to attack the part of the tumor that's growing. And so you could think of this as not only a clinical problem, but potentially an interesting scientific opportunity where everyone here knows that gastrointestinal stromal tumors are caused by somatic mutations in c-KIT. And could new mutations have evolved over the course of Gleevec treatment or his various, the treatments that he received that are causing this bit of growing tumor to evade the treatment with the sorafenib? And so if he did, so the idea is if he did develop these exon 13, exon 17 mutations, we know that those are susceptible to a different tyrosine kinase inhibitors. Exon 13 and exon 17 shown here. If he had those, we could switch him to a drug that should work against those mutations, dasatinib. Alternatively, it's known that the PI3 kinase AKT pathway can be a survival pathway, and so we contacted Gordon Mills, who does phosphoproteomics at MD Anderson, and would he be interested in trying to help us understand what the differences in signaling are between the growing bit versus stable bit of disease, and could that allow us to try to identify a vulnerability? And the interesting thing is, scientists, hardcore scientists who are very, kind of very immersed in what they do, I think can become interested in this type of topic if they think that there's a real chance to make an interpretation, a valid interpretation from their findings. So this was one early example. As it turned out, the patient came here to the university. We biopsied we means the surgeons biopsied the liver. It turned out not to be tumor in his liver, but just fat. And so we didn't end up getting an answer from this. It turned out that he didn't have growing disease in the liver. About 6 months ago, people in this room will recognize this person, Paul Ramsey, our dean, asked me to bring this concept of partners in personal oncology into the university. And I've been working on this for the last few months, and we're creating a center called the Center for Cancer Innovation. And around that concept, I met with a number of faculty. We had our first meeting in September on a Sunday night at 8:30 p.m. and 18 faculty members came. A bunch of people from genome sciences, a bunch of people from oncology. The genome sciences people were amazed that there were so many oncologists there, and the oncologists were impressed that there were so many genome scientists there. And to date now we have more than 50 people from various institutions, the university, Fred Hutchinson, Institute for Systems Biology, SAGE, and as well as oncologists in private practice. And around this effort we have now 3 teams that have developed. And these teams are all working around the concept of enrolling our first patient by June 3rd of this year. These lists are probably not entirely up to date, but these are the 3 teams that now meet regularly around the goal of getting our first patient enrolled. Now, what would be a reasonable way to go about doing this? So I'll just share with you what I think is our latest idea about what a reasonable approach would be and what a reasonable target disease might be. And I think the initial focus for us will be in triple-negative breast cancer. And part of the idea here is that obviously breast cancer is common. There are many patients with this problem. There's a large number of drugs that are available and a lot of investigational drugs in breast cancer. If you think about it, how would we take a patient and do this kind of extensive analysis and make a recommendation back to that patient? How could we try to do this in a way that's not just ad hoc and not just picking something that looks attractive in a kind of an unsystematized way? And should we focus our efforts on single gene mutations where it's pretty straightforward? If you have an ALK mutation, you'll respond to one drug. If you have an EGFR mutation, you'll respond to another. And I think the problem with focusing on single gene mutations is that they are relatively rare. If all we do is based on trying to look at individual genes, we'll only be able to make useful recommendations, I think, in a small fraction of patients. And so the way that we're looking to do this initially is based on gene expression profiling, combined with exome sequencing, combined with some type of proteomic analysis. But our initial work will probably rely on exome sequencing and RNA sequencing. The approach we're going to take is built off of work that's described not only in this paper, but this paper provides, I think, an important example of the approach that we're planning to take, where breast cancer can be subgrouped into different types based on gene expression profiles. And if you take all breast cancers, there are very well-defined groups that can be identified by various clustering algorithms using microarrays. This paper described attempts to cluster breast cancers only within the group of patients with so-called triple-negative breast cancer. And triple-negative breast cancer accounts for about 15% of breast cancer. Since it's negative for HER2 and negative for estrogen receptor, there aren't good targets for treating it, and it tends to be an extremely difficult problem. And it's a disease where new treatments are urgently needed. And so this paper described an effort to subgroup patients with triple-negative breast cancer based on their gene expression profiles. And there are various subgroups that these investigators came up with. One that I think is interesting, especially interesting, is this is clustering from a training set of patients and then a validation set of patients. But what they found that was interesting, and this isn't entirely new information by any means, but they found that about 10% of patients with triple-negative breast cancer have a gene expression profile that that looks like a response to androgen receptor signaling. And so the notion that about 10% of patients with triple-negative breast cancer may have signaling driven through androgen receptors. Interesting, of course, because there are lots of drugs that are used in the world of prostate cancer to interrupt androgen receptor signaling. And so that's interesting. There are other groups that have that seem to be driven by other signaling pathways. Epidermal-derived growth factor receptor is expressed in about half of patients with triple-negative breast cancer, have EGF receptor expression by IHC, and there's a significant EGF receptor signature within this group of patients that's shown in some of these other subgroups. Brig Meekum, a scientist at Sage Bionetworks here in Seattle, used the gene expression profile for androgen receptor signaling and looked across patients. So the nice thing about Sage is that they have access to tens of thousands or hundreds of thousands of gene expression signatures that are publicly available. And they can interrogate all of the publicly available data. And what Brig found was that if you look at, if you generate an androgen response signature and you look for its presence across thousands of patients with cancer where gene expression profiles are now in databases, what Brig found was that unsurprisingly, the most abundant androgen, or the most prominent androgen response signature, occurs in patients with prostate cancer. But he also found that there are patients with breast cancer who appear to have tumors with this androgen response signature. And so the way that we're thinking about doing our first trial shown here, where, and this, I should say, hasn't been completely signed off on, by all of the subgroups of our center, and especially the clinical teams looking this over. And really, I'm trying to just invite criticism, because I think we need to make sure this is as good as it can possibly be. But the idea is to have a patient with metastatic triple-negative breast cancer. And again, these will be patients who are highly selected based on having disease that we think is amenable to repeated biopsies, and who are, kind of psychologically enthusiastic about participating in this trial and understanding the limitations of the state of knowledge right now. So patients with metastatic triple-negative breast cancer, do a biopsy, give everyone after this first biopsy a drug, and the same drug, and I'll tell you what that drug is in a minute. When the disease becomes resistant or recurs, do a second biopsy, and then assign initial treatment not based on the results of omics studies, but on the basis of studies that pathologists do standardly, just immunohistochemistry, for androgen receptor, EGF receptor, which is supposed to occur in about 50% of patients with triple-negative breast cancer, HER2, in case you missed them previously, or tumor may have changed, or estrogen receptor, progesterone receptor, or there would be a group that's IHC negative. And the initial drug that we want to use is a drug that inhibits the mTOR signaling pathway. And the reason for that is that in the Cancer Genome Atlas, a paper in Nature from about 10 weeks ago, characterized the genetic profiles of patients with breast cancer generally, and within the patients with triple-negative breast cancer, there was an especially prominent signal for the PI3 kinase AKT-mTOR pathway. And so this pathway seems to be prevalent in patients with triple-negative breast cancer. And so give them initially a drug that blocks the mTOR pathway, And then when they either become resistant to that or shown to be resistant to that or respond and then recur, we do a second set of biopsies, and then we assign them to treatment according to immunohistochemistry, depending on these different groups. And if they have androgen receptor positive disease, we add, on top of the everolimus, we add an androgen receptor blocker, bicalutamide. If they have EGFR positivity, we keep them on everolimus and we add an EGFR inhibitor, lapatinib. Lapatinib also works with HER2, so we could add, for patients with HER2 positive disease, we'd add lapatinib. Or if they have estrogen receptor or progesterone receptor, we add exemestane. And actually, this combination of everolimus and exemestane is already FDA approved for ER positive, estrogen receptor positive breast cancer. And I should say all of these combinations, these drug combinations of everolimus and bicalutamide, lapatinib, and exemestane have been tested in patients. They're not, and they've been tested in clinical trials. So there's some sense of what the dose and schedule of these combinations would be within individuals. And then if they're IHC negative, we would add carboplatin because of the notion that triple negative breast cancer, a substantial subgroup of those patients have gene expression profiles that's reminiscent of serous ovarian cancer, and there's growing interest in using platinum-based drugs or PARP inhibitors in patients with triple negative breast cancer. And then when the patients develop resistant disease or recur, then we would do another biopsy. And from each of these biopsies then do our omic analysis and come with a recommendation that would lead to a customized treatment in that patient. And then this longitudinal effort to treat, observe, measure, treat again, and try to develop this longitudinal experience. So this is what I imagine our first generation trial will look like. What I think this would allow us to do that's really compelling to me in our second generation trial is shown here, where we have, again, the patient. This is very similar to what I've shown you already. We have the patient at the beginning, we do the biopsy, we give the first drug, we do the rebiopsy. But now if you think that we're looking at the mTOR signaling pathway and we're giving everolimus to block the mTOR pathway, actually quite a lot's known about mechanisms that tumors can use to escape the mTOR pathway. One, and what we could do is we could focus on the key thing that I want to draw your attention to here is the delta. So instead of comparing this patient at this point in time, so instead of saying, okay, we're going to biopsy this patient here and only compare that patient to all prior knowledge, so all other patients with cancer, and try to figure out where does this patient fit into the universe of cancer cases that have been studied so far. The idea here is we could compare what happened, how did the patient respond to this intervention, to this biological perturbation of their tumor, and comparing the rebiopsy with the initial biopsy. And so it's known, for example, that when you block the mTOR pathway, one way of circumventing that is to upregulate PI3 kinase signaling. And you could say, okay, if the patient has upregulation of PI3 kinase signaling, we're going to give a drug, an investigational drug, add an investigational drug that blocks PI3 kinase. If they have an increase in MAP kinase signaling, we're going to add a drug that blocks that. And if they, and now just last month there was the notion that JAK2 signaling might be responsible for resistance to mTOR inhibition. And so you could think about then adding a JAK2 inhibitor that's now FDA approved. So I don't have a concluding slide. This is my concluding slide, and I'd be happy to answer any questions.